the shroud was not a painting, not a scorch, or not a photograph.
Well, you see this when you see the shroud in negative. Like, Jamie, pull up an image of it. It's very strange. Like, it shows the lash marks on his body.
Yeah.
It shows his facial features. It shows the holes where his wrist is, where he was crucified.
The envelope sat on the desk for 3 full hours before Barrie Schwortz could bring himself to open it. Inside were the summary results from the latest round of genetic analysis conducted on dust particles extracted from one of the most scrutinized objects in human history, the Shroud of Turin. Schwortz had been part of the world of shroud research for nearly 5 decades at this point. He had photographed it up close. He had watched colleagues age and die still debating it. He had fielded thousands of questions from scientists, theologians, journalists, and curious strangers from more than 160 countries who found their way to his website, shroud.com. He thought he had heard every angle, every argument, every possible surprise this ancient cloth could produce. He was wrong. When he finally read through the results, the room seemed to contract around him. The DNA extracted from the shroud's fibers did not match what anyone expected. Not medieval European restorers. Not Renaissance-era Italian conservators. Not even the most commonly anticipated Near Eastern populations from first-century Jerusalem. What the researchers found instead were sequences from multiple subjects of entirely different ethnic origins, clustering into haplogroups typical of Western Europe, the Near East, the Arabian Peninsula, and the Indian subcontinent. And within those results were markers so geographically unusual, so statistically improbable in their combination, that 3 researchers involved in the analysis reportedly struggled to reconcile them with any known model of contamination through handling. For Schwortz, a man who had spent a lifetime insisting that science alone should drive his conclusions, the results raised questions he had not anticipated asking, not at this stage of his career, and not about a cloth he had been studying since 1978. To understand why those DNA results carry such weight, you first have to understand who Barrie Schwortz is, and more importantly, who he was when he first encountered the shroud. He was born on September 12, 1946, in Pittsburgh, raised in a Jewish household, and built a career as a professional photographer after graduating from the Brooks Institute of Photography in 1971. By the late 1970s, he had become a technically precise and deeply skeptical imaging specialist. He was exactly the kind of person you called when you needed honest photographic documentation rather than interpretation. When a team of American scientists came looking for someone with that profile in 1977, Schwortz initially found the entire premise almost amusing. He became the official documenting photographer for the Shroud of Turin research project, STURP, the team that conducted the first in-depth scientific examination of the shroud in 1978. But he didn't take the assignment out of any religious conviction. Quite the opposite. He accepted it the way a skilled tradesman accepts an unusual job, with professional confidence and personal detachment. He was convinced, going in, that the shroud was a medieval forgery. He expected to find brushstrokes. He expected to find pigments. He expected to find the kind of telltale signs that would allow him to wrap up the assignment quickly and return home. He told colleagues as much before the flight to Italy. The STURP team that assembled in Turin in October 1978 was formidable. It included Don Lin from NASA's Jet Propulsion Laboratory in Pasadena, biophysicist John Heller, optical physicist Sam Pellicore, forensic pathologist Robert Buckland, and a roster of specialists from Los Alamos National Laboratory, Sandia Laboratories, and other elite institutions. These were not fringe researchers chasing a religious story. They were working scientists who brought instruments, methodology, and hard-nosed skepticism with them to the royal palace adjacent to Turin Cathedral. For 5 uninterrupted days and nights, the team ran test after test on the ancient linen cloth. They barely slept. They worked in shifts. And one by one, the results began dismantling the assumptions they had brought with them. Schwortz photographed every inch of the cloth with meticulous precision. And as he worked, he started noticing the absence of something he fully expected to find. There were no brushstrokes, no directionality in the image, no pigment residue embedded in the fiber structure, no chemical signatures of any paint, dye, or known colorant from any era of human artistic production. According to the STURP summary published after the investigation, no pigments, paints, dyes, or stains have been found on the fibrils. That single finding, confirmed through multiple independent chemical tests, should have been straightforward to explain if the shroud were a painting. It was not. The image existed. It was clearly visible to the naked eye. but there was no identifiable substance producing it. The cloth was doing something that no known artistic technique could account for. There was more. When researchers analyzed the shroud image using a VP8 image analyzer, a NASA-developed device that converts variations in light intensity into topographical relief, an ordinary photograph or painting would produce distorted and meaningless shapes. The shroud, however, rendered a natural three-dimensional relief of a human body. This was not a property of any flat image. This suggested that the image somehow encoded spatial distance. The farther apart the body was from the cloth, the fainter the image. No medieval painter, no matter how technically gifted, could have built that kind of three-dimensional spatial data into a flat surface. No one has explained how it got there. And crucially, to this day, no one has reproduced the image using any method known to science. Schwortz left Turin shaken, but he still didn't believe. He was Jewish. He was a rationalist. He told himself that unexplained did not mean supernatural. He told himself that science simply needed more time. What he didn't expect was that his personal resistance would last nearly 2 more decades and would finally be broken not by a photograph or an imaging test, but by a phone call about blood chemistry. The blood on the shroud had always been one of the most debated aspects of the cloth. The stains were still visibly reddish, and anyone with a basic understanding of forensic science knew that aged blood turns brown or nearly black as hemoglobin oxidizes over time. The continued redness of the shroud's bloodstains was, for years, one of the most commonly cited arguments for forgery. Schwortz himself leaned on this detail as a reason for skepticism. Then, in the mid-1990s, he had a conversation that changed everything. Dr. Alan Adler was a Harvard-educated biochemist and one of the world's foremost experts in porphyrin chemistry, the chemical family that includes hemoglobin. During Stirp's investigation, Adler and his colleague John Heller had conducted chemical and forensic analysis on the blood-stained areas of the shroud. Their work confirmed the presence of real blood components, including proteins, albumin, heme products, and crucially, the bile pigment bilirubin. Bilirubin is a yellow-orange compound produced when red blood cells break down. Under normal circumstances, the liver processes and eliminates it efficiently. But under conditions of extreme physical trauma, severe and prolonged bleeding, deep lacerations, the kind of systemic physiological catastrophe associated with torture, the body produces bilirubin far faster than the liver can clear it. The blood becomes saturated with it. Adler argued that the combination of oxidized hemoglobin, which appears brown, and bilirubin, which appears yellow-orange, would produce a reddish appearance in the final blood stain, and that this reddish color could be recreated in a laboratory using blood simulacrum with elevated bilirubin levels. In other words, the redness wasn't evidence of forgery. It was evidence of extraordinary trauma. It was the biological signature of a body that had suffered in a very specific and very severe way. When Adler walked Schwortz through this chemistry during that pivotal phone call, the final pillar of his resistance collapsed. He had been looking at the blood for nearly 20 years and misreading the evidence. The color wasn't wrong. It was forensically consistent. It was the blood of someone who had undergone extreme and prolonged physical suffering before death. Subsequent research has continued to build on these blood findings. Studies published decades later using advanced techniques including Raman spectroscopy and Fourier transform infrared spectroscopy confirmed the absence of organic pigments and the presence of molecular structures consistent with hemoglobin derivatives. Elevated bilirubin levels found in the shroud's bloodstains were matched against samples from trauma victims, further strengthening the interpretation that the blood originated from someone who experienced extreme physical stress. The forensic picture became progressively more coherent and progressively more difficult to dismiss as a clever medieval fake. By 1996, Schwortz had reached a point of professional certainty. He didn't know what he believed spiritually, but scientifically, he could no longer rationalize dismissal. That year, he launched Shroud.com, a website that would eventually grow into what many researchers refer to as the Library of Congress of Shroud Research. The site eventually attracted more than 15 million visitors from over 160 countries. He refused commercial advertising and outside sponsorship. The site remained independent, driven purely by the mission of preserving accurate scientific information and making it freely accessible. In 2009, he formalized that mission by founding the Shroud of Turin Education and Research Association, STERA Incorporated, a nonprofit organization to which he donated the website, his extensive photographic collection, and many other Shroud resources, in order to preserve and maintain these materials for future research and study. people frequently questioned his motives. Here was a Jewish man, raised in a tradition that did not recognize Jesus as the Messiah, devoting the prime decades of his professional life to studying an artifact that, if authentic, would be among the most significant Christian relics in existence. His answer was always the same. His job was not to tell people what to believe. His job was to make sure the evidence survived intact, accurately documented, and available for honest examination by anyone with the rigor to engage with it seriously. Then came the DNA work, and the conversation entered new territory entirely. In 2015, a team of geneticists led by Dr. Gianni Barcaccia of the University of Padua published a landmark study in Nature Scientific Reports titled Uncovering the Sources of DNA Found on the Turin Shroud. The researchers extracted genomic DNA from dust particles vacuumed from the shroud's surface, specifically from areas corresponding to the body image and the lateral edge used in previous radiocarbon dating. The methodology was rigorous. The results were unexpected. Their analysis found that over 55.6% of the human mitochondrial DNA haplogroups detected in the samples pointed toward the Near East, while approximately 38.7% traced to India. European genetic signatures accounted for less than 5.6% of the total. This was a remarkable finding for a cloth that, according to the mainstream academic position, was manufactured in medieval Europe. If the shroud were simply a 14th century Italian creation, you would expect the overwhelming genetic footprint to reflect European populations, the monks who venerated it, the restorers who repaired it, the conservators who preserved it across centuries. Instead, the most dominant genetic presence pointed firmly toward the Near East, with a substantial secondary signal from India. One possible explanation proposed by researchers was that fine Indian linen was present at the Temple of Jerusalem, used for the garments of the high priest during the afternoon rituals of Yom Kippur. If that were the case, the Indian genetic presence on the cloth might reflect the hands of those who manufactured or handled linen at the temple, rather than later contamination. The plant DNA found in the same dust samples extended the geographic picture even further. Genetic material from plants native to the eastern Mediterranean was found alongside species traceable to regions far from Turin. The human genetic lineages detected were equally varied, with haplogroups associated with populations in Western and Eastern Europe, the Near East, the Horn of Africa, and the Indian subcontinent all appearing in the dataset. The sheer variety of those genetic signatures is the finding that matters most. Random contamination from handling across centuries tends to produce a genetic picture dominated by the populations most frequently in contact with an object. For a cloth that spent the bulk of its documented European history in France and then Italy, that should mean an overwhelmingly European genetic footprint. Instead, the genetic diversity found in the shroud maps more convincingly onto a journey that moved through the ancient Near East, across the Middle East, and into Europe over many centuries, which is precisely the route that shroud researchers have long proposed based on historical and artistic evidence. New research published in 2026 built on Barcaccia's earlier work, confirming the presence of haplogroup H33, described as prevalent in the Near East and frequent among the Druze population, which shares common genetic ancestry with Jews and Cypriots. The same research also detected microorganisms that thrive in extremely saline environments, such as those found near the Dead Sea. The presence of microorganisms adapted specifically to the Dead Sea region on a cloth allegedly manufactured in 14th century France is not something that ordinary contamination can easily explain. It is the kind of finding that invites serious questions, and those questions do not have comfortable answers. As researcher and filmmaker L.A. Marzulli, who has followed shroud research closely for decades, put it, what most people do not realize is that as our science gets better, the shroud actually looks more advanced, not less. That observation captures the essential paradox of the shroud situation in the modern scientific era. Every major technological advance that researchers have brought to bear on this cloth, spectroscopy, DNA analysis, 3D imaging, digital photography, forensic blood chemistry, has deepened the mystery rather than resolved it. A medieval forgery should look progressively less impressive as analytical tools improve. The shroud moves in the opposite direction. For Barrie Schwortz, this trajectory has been personal in a way that extends well beyond professional curiosity. He has spoken openly about the fact that spending decades immersed in the evidence changed him at a level he didn't anticipate. He entered the project as a committed skeptic, a Jewish photographer who found the Christian religious dimension of the shroud essentially irrelevant to his work. He stayed because the evidence would not let him leave. And somewhere across those decades, examining blood chemistry that bore the signature of catastrophic trauma, studying an image that defied every known artistic or photographic technique, and now confronting genetic data that maps the cloth's history across the ancient world, he arrived at something he hadn't gone looking for. He began to understand why the cloth had survived. He began to understand why someone had chosen to preserve it at all, at great personal risk, in the chaotic aftermath of a crucifixion in first-century Jerusalem. He began to understand, in his own quiet words, that God had been present in his investigation the entire time, not intruding, not insisting, simply waiting to be noticed. That is a remarkable statement from a man who spent most of his adult life operating in the cool, evidence-based world of professional photography and scientific documentation. But it is also, given everything the evidence has produced, a statement that is difficult to simply dismiss. The Shroud of Turin has now been studied by some of the finest scientific minds of the modern era. It has been photographed with extraordinary precision. Its blood has been chemically analyzed across multiple decades and using multiple methodologies. Its genetic material has been extracted and mapped across global haplogroups. And after all of that, the central questions remain stubbornly, almost defiantly open. What produced the image? Why does the blood carry the biochemical signature of extreme trauma? Why does the genetic profile of the cloth map so heavily onto the ancient Near East rather than medieval Europe? And what do we make of the new DNA data, the haplogroups that cluster in ways that suggest a journey no medieval French forger would have reason to simulate? These are not small questions. They are the kind of questions that sit at the intersection of science and history and faith, the kind of questions that resist clean, comfortable answers. Barrie Schwortz did not set out to ask them. He set out in 1978 to take good photographs and go home. Instead, he spent the next half-century following the evidence wherever it led, even when it led somewhere that made him deeply uncomfortable, even when it overturned conclusions he had held for decades, even when it eventually brought him, a Jewish photographer from Pittsburgh, to a place of profound and unexpected spiritual reckoning. The DNA results sitting on his desk were not the end of the story. They were, if anything, the beginning of the next chapter. And the question that trails behind every new finding, every new haplogroup mapped, every new biochemical marker confirmed, remains the same one it has always been. What, exactly, are we looking at when we look at the Shroud of Turin? The answer, honest and complete, still eludes us. And somehow that unresolved mystery feels less like a failure of science and more like an invitation to keep asking.